Welcome to Cube Talks, Discovery Science Series presented virtually by Cube's Innovators every second Tuesday of the month. We are very pleased to welcome Dr. Amy Zimmer Faust, who will be speaking for 20 minutes and taking questions on the scoop on poop, pollutants, and water quality. Thank you for joining each month to learn more about and embrace science in our lives. In the upcoming months, we are enthusiastic about sharing experts in science that help make our planet a healthier place. We have an exciting schedule coming up. On December 8th, we're going to welcome Dr. Dan Cooper, who's going to talk about COVID and kids. He's a professor of pediatrics at UCI's Institute of Immunology. On January 12th, we're going to dare mighty things, Mars rovers paving the way, with Nikki Cox, and learning more about that incredible team. And February 9th, ankle biters, invasive mosquitoes with OC vector control. The housekeeping rules are pretty simple. In order to not distract from the presentation, everyone will be muted until our question and answers. If you have a question, please save them for this period of time and raise your hand to be called on. We will give you limited amount of time for each person to ask questions just so we can cover all the questions that we might have, but we want as many as possible because if we're not able to get them all, we'll make sure that we follow up with any answers. Of course, it goes without saying, we ask everyone to be kind and respectful during any and all communication. And so, we could actually mute you or eject you if you get too crazy or do any Zoom bombing. So just know that. Any additional technical questions or questions not related to the presentation, please directly message to me or to Elizabeth. As always, have fun, be curious, and make friends. For those who were unable to join us or would like to view this broadcast at a later date, we will be recording it and making it available on YouTube. Please allow me now to welcome Lisanne Sebastian, a member of our Discovery Cube's Innovators and Leadership Council, as well as Janet and Waukie's daughter, Lisanne.
Hello, welcome to the Innovators Cube Talk. I'd like to introduce Amy Zimmer Faust, a friend of mine, a fellow paddler and a science nerd. Amy Zimmer Faust is a research scientist with experience working at local and federal public agencies. She currently works at the Southern California Coastal Water Research Project, a public agency that conducts environmental research and was previously at the US EPA. Her research is primarily focused on water and health and the relationship between the environment and the sources, occurrences, and fate of waterborne contaminants. She is a SoCal native, receiving her PhD in environmental science and engineering from UCLA and her BS in aquatic biology from UCSB. Please welcome Dr. Amy Zimmer Faust.
All right. Yay. Thanks, Lisanne, for the introduction. So let me make sure that you can all see my slides okay. Yeah?
Yes.
All right, and I will jump right in. So today I'll be talking a little bit about pollution in the coastal environment, how it impacts public health, where it comes from, and then how we can better manage for it. So Lisanne already gave a bit of an introduction about me and kind of my background, but I do want to talk briefly about how I kind of ended up studying water and health. So I got my start studying marine biology at UCSB. And I was a surfer and involved in Surf Rider, and I kind of wanted to save the waves. So I got my start looking at systems that look a lot like this. But I became increasingly interested in the intersection between public health and water quality. And when you hear of that, you probably think of drinking water. But there are also lots of ways that exposures in recreational waters can actually impact public health as well, which is what I went on to study at UCLA, where my study sites unfortunately started to look a lot more like this. So since then, I've been lucky enough to work as a researcher at both the EPA and currently I'm with SQUIRP, where I've looked at all kinds of coastal water exposures and pollutant sources, including agricultural runoff, stormwater, and wastewater. So what is SQUIRP? So as Lisanne mentioned, SQUIRP stands for the Southern California Coastal Water Research Project, and we are located right in your own backyard in Costa Mesa. And even though project is in the name, we've actually been around for 50 years now. We kind of end with project, and that's a result of that kind of reflects the history of SQUIRP. We were founded as a result of the Porter-Cologne Act, which was passed in 1969. And the Porter-Cologne Act is California's own Clean Water Act that actually predates the federal Clean Water Act. So the Porter-Cologne Act was passed, and regulators and dischargers alike really didn't know what to do about it and how to kind of manage and regulate and implement it. So SQUIRP was founded as a result to kind of provide the neutral science that could help managers kind of fill in data gaps and actually apply the Porter-Cologne Act for improved water quality. So we have a commission, and we've had a commission for our entire 50 years that's comprised of both regulators and regulated. So the regulators, those are the people in charge of coming up with and enforcing water quality standards in California. That's the EPA, that's the state water boards and folks like that. And then the regulated, those are the dischargers that actually have to comply with the water quality standards. So this includes the sanitation districts, this includes the public utilities that are discharging things like wastewater and stormwater to our coastal systems. So SQUIRP is actually made up about 50 scientists, and we hear from our commission about what science and data is needed to manage coastal water quality routinely. And then we go out and we conduct that science, and then we brief the managers. So it's this really nice kind of network of folks that can implement the science and the scientists that can do the science that's resulted in some pretty neat impacts to water quality. So today what I want to do is kind of give you a brief overview of beach water quality and how we monitor beach water quality and what we actually measure. And then I want to talk about new technologies that are helping us to improve this monitoring. And then I'll talk about a few examples that are kind of right in your own backyard about how we actually find and fix water quality problems for public health protection. And then I'll end talking a bit about where the science is all headed. So in California, you know, we really love our beaches, and it's a huge part of our culture and economy. And I know that I rely heavily on the ocean for my own sanity, especially in the last 6 to 8 months for COVID reasons. And as a result, California does invest a lot into monitoring. We've made significant improvements in beach water quality in general in the last 20 years or so because of all of these investments. However, many challenges still exist. We still have beach closures from pollution events. We still have lots of incidences of illnesses related to exposures that happen when people are recreating in our beach waters. So these illnesses can occur from swimming. These illnesses that do occur from swimming in polluted water are a result of lots of different pathogens that come from lots of different sources. And some of these probably look familiar, maybe not look familiar, but sound familiar, like norovirus and salmonella. And maybe first you think of contaminated lettuce or something like that. But these are pathogens that are also associated with illnesses from recreational water exposures as well. And they cause some not very fun symptoms. And these presence of these pathogens is actually the reason that we have beach closures and monitoring, so that we can try to reduce the amount of times that people are swimming and surfing and paddling and whatnot when water quality is bad and could actually make you sick. So when you see a beach closure sign, however, that closure sign is not actually based on measurements of those pathogens that I just mentioned, like salmonella and norovirus, primarily because these things are really hard to measure, the pathogens that make you sick, due to the fact that they occur at such low concentrations in the environment. So instead, we actually measure bacteria that are associated with fecal sources and indicate the presence of those pathogens that we worry about. So these indicator bacteria are present at much higher concentrations and are not human pathogens, so making them much easier and safer to measure. And these general fecal indicator bacteria are associated with lots of different types of poop. Birds, cows, people, all kinds of things. And levels of these bacteria, when they're high, beaches will be closed because they're indicating they're indicating and reflective of the presence of the pathogens that actually do make us sick and that we worry about. And different public health agencies all over the coast are responsible for routinely monitoring for these types of bacteria. And this data is publicly available. So you actually have an Orange County Beach Info website. I've just shown today here, which is all yellow because it just rained. So there's a rain advisory in effect right now, where these general fecal indicator bacteria data are posted online and then updated weekly. So you can go online if you're interested and see how your local water quality is looking. And if you should be snorkeling or whatnot, or maybe holding off for a few days. So indicator bacteria are really useful for giving us kind of a general idea of if water quality is bad. But as I mentioned, they don't give us any information about the source of the pollution. And we care about the sources for a couple of reasons. One, because public health risk does vary by source. For instance, sewage is much more likely to make you sick than poop from seagulls. And although there are pathogens in animal poop that can make people sick, the risk is typically much lower. It's also really hard to fix a problem if you don't know what it is or where it's coming from. And there's new technologies that we use as kind of water quality scientists to actually identify the source of contamination. And these methods are primarily based on looking at the DNA from bacteria in beach water samples. So what we do is we take a water sample that's full of bacteria, both good bacteria and bad bacteria, and we isolate and extract all of the DNA that came from bacteria in that sample. And different kinds of bacteria actually come from different types of poop. So we can take advantage of this and look for DNA from those specific types of bacteria. For instance, we could look for DNA, pretend this is a DNA sequence. So we could look for DNA from bacteria that comes from human poop. Or we could look for this sequence that could be coming from DNA that comes from bacteria associated with gull poop. So there's been a lot of research in this field to get this kind of library of DNA sequences that we know come from all of our usual suspects that are all impacting water quality, leading to elevated bacteria levels. So this includes wildlife, agricultural animals, and humans. So basically, to summarize, with this method, we're just looking for and measuring specific DNA sequences that are associated with these different types of poop. It's kind of like CSI for poop, for all of the types of poop that we worry about at our beaches. But more recently, we actually have even newer methods that are also kind of based on the same idea of taking a water sample and extracting all of the DNA and analyzing it. But in this case, we're not just looking for one sequence that's associated with a specific source. We're actually getting an idea, gaining an understanding of all of the types of bacteria that are present in that sample. So if you see this colored bar, you can imagine that each color represents a different type of bacteria. So here, we've generated kind of this unique fingerprint for our water sample that's telling us about all of the bacteria that are there and how abundant they are. And we can do the same thing with different sources. We can create these unique fingerprints of all the different types of bacteria that are present in all of our sources. And then we can look for those fingerprints in our water sample. So to summarize, we kind of have two fancy ways that are based on DNA, where we can measure. The first was we can measure specific DNA sequences that we've matched to particular sources. And the second, this more recent method, we can actually create these kind of bacterial fingerprints for any source we want and then look for those. So with that, hopefully I haven't confused you too much. I'm going to switch gears and talk about two projects where we use these types of techniques to find and fix particular water quality issues. The first being in Topanga Canyon. So Topanga, for those of you not familiar, probably most of you are, it's located just north of LA. It's got some fun little waves, a very good Thai restaurant, Cholada Thai, and some great hiking. So not a bad place to do some field work. But it also has a history of really poor water quality and beach closures without a lot of success in terms of finding and fixing and understanding what's causing those problems. So I was part of a big collaborative effort where we sampled in Topanga Lagoon, we sampled the beach, and then we sampled Topanga Creek, which flows all the way from the town of Topanga to the lagoon. And we were looking for different sources and trying to get an idea of what's leading to elevated bacteria levels at the beach. So after some initial sampling, we had some interesting trends. First, we had high bacteria levels shown with these red numbers. Those are really high. Probably don't want to go surfing if levels are this high. So we had really high levels at the beach and in the lagoon, which wasn't surprising. We knew this was a problematic site. But then we had this second interesting thing where we also had high levels of pollution just coming from the town, but these really weren't transported very far within the stream. So this was really interesting, and we were curious, where is the pollution from the town? Where is it going? So we have these two questions. First, what is leading to elevated bacteria levels at the beach? And then where is the elevated bacteria in the town going? So two main questions. So first, I'll focus on what's going on the beach and the lagoon. So here we measured two of our usual suspects. We measured elevated levels of that DNA sequence associated with dog poop, and we measured that DNA sequence associated with elevated levels of gull poop. And we saw some really interesting seasonal trends. With the gull poop, regardless of if it was winter or summer, we saw really high levels in the lagoon, and that's where all the gulls like to hang out, and then high levels, but not quite as high in the beach. So this is definitely kind of one of the sources of these elevated bacteria levels at Topanga. What was interesting about the dog marker is that we had really high levels in the winter and really, really low levels in the summer. And what we think is going on is that in the summer, as we all know, there's lifeguards, there's increased enforcement and social pressures to probably not have your dog on the beach. And if it is there, probably pick up the poop. And the winter, this enforcement goes away, we see the dog marker levels increase, and we actually saw an impact on water quality. So I know that I'm definitely guilty of kind of knowing the lifeguard schedule and taking my dog to the beach around it. So if you're doing that, just make sure you're picking up after your dog, because otherwise its poop might become part of a study like this. Probably don't want that. All right, and then the other interesting thing about Topanga was that in the upper watershed, we saw that the pollution really wasn't making it very far. So what we did is we took water and sediment samples. We did a bunch of laboratory experiments looking at kind of properties of the water and sediment to see what's going on. So using the data from those experiments, created these models that took into account kind of all of the physical and biological things that are affecting pollution within that stream reach. Don't worry about looking at this too closely. It's just that there are lots of things that are impacting pollution. And we found that that stretch of the stream shown here is actually pretty unique and has some properties that make it a lot like a natural water filter. A few of those things are that it's not very steep, so it's kind of flat. The water really slows down through this stream reach, and it's really sunny. So within Topanga Creek, the rest of the creek is pretty shaded. This is really sunny and has really low flow rates. So you can imagine it's kind of acting as this natural water filter. Pollution's flowing in from the town, but there's actually time for things to settle in and times for the sun to kind of zap all the bacteria and things that we worry about. So it's almost like you have this natural UV water filter occurring just below the town of Topanga. Lucky for them. So what came from this is that there was actually increased enforcement of the no dog policies in the winter and increased education around picking up dog poop. And then we also made recommendations to really double down and focus on lower watershed sources, seeing as they had this kind of natural filter between sources from the upper watershed and the beach. So next, I want to talk about another project briefly that's ongoing near the US-Mexico border. So you may have seen headlines like this. We know that the border region has kind of a history of poor water quality, leading to really frequent closures of San Diego beaches in this region. So in fact, on average, beaches located near the border are closed about 100 days a year. So that's a lot, about a third of the time. In addition, surfers and lifeguards frequently report kind of sewage odors while recreating without knowing what the source is. So this has fueled a lot of media coverage with people convinced that these sewage spills are a result of sewage discharges originating in Mexico. So there's kind of two primary sources within this region that may be to blame and that kind of get the most attention. The first being the Tijuana River. The Tijuana River crosses the US-Mexico border, with a large part of it is actually located in Mexico, and receives a lot of sewage inputs on the Mexico side of the border. And we know that in Tijuana, this is kind of a result of the fact that in Tijuana, the drainage system is not actually capable of kind of capturing, holding, and treating all of the sewage that it generates. And this is due to a lot of issues, including aging infrastructure, overburdened wastewater treatment plants in Tijuana, and a large population living in the region without access to any kind of sewer infrastructure. So we know in wet weather that this is a big source of kind of pollution within this region, that there's this overflow into the Tijuana River, comes across the US border, and it spills out into the ocean. In dry weather, flows before it gets to the US side are actually supposed to be diverted to different wastewater treatment plants. So in dry weather, it's not supposed to be as much of a source to kind of water quality issues within this region. So first we have the Tijuana River that gets sewage from lots of different things in Mexico, crosses the border, discharges right just south of Imperial Beach. So that's potential source number one. Potential source number two, and you may have seen videos or footage of this in the news, is this wastewater treatment plant that discharges at Punta Bandera. So this is located about 6 miles south of the border. And this wastewater treatment plant treats about 25 million gallons a day of wastewater, which is equivalent to 40 Olympic-sized swimming pools a day. So this is a lot of sewage. And this is like very minimally treated sewage. So it's kind of settled and then it's dumped into the ocean 6 miles south of the border. So there's been some modeling that's been done by folks at Scripps in UC San Diego, showing that this plume from this plant could travel all the way north and impact beach water quality in San Diego. But up to this point, we haven't had actual kind of measurements to validate this work. So what did we do? Well, we conducted a bunch of targeted sampling in this region, kind of in collaboration with those folks at Scripps who are doing the modeling, to see if in fact that plume from that wastewater treatment plant could make it all the way up and impact beach water quality in San Diego. So we conducted sampling during very specific swell and wind conditions when we think that currents are moving from the south to the north, to get an idea of just how far north this plume can travel on kind of these like best conditions for this type of impact. So we completed targeted sampling from just south of that sewage plant all the way to Silver Strand State Beach, which is about a 14-mile stretch, with the idea being that we would look for a gradient in pollution moving north away from the plant. So the plant is here. So we were sampling from about here all the way north. And then remember, in kind of closer to the beaches that we were really looking at and investigating, we also have this other potential source, which is the Tijuana River. We've got two things going on, that wastewater treatment plant and then the Tijuana River. So what were we looking for? Well, we were collecting samples and we looked for the DNA sequences that I talked about associated with human sources to see if there was a gradient as we moved north away from the plant. But one thing about kind of this method is that this tells you if there's human fecal pollution in the water, you can look for a gradient. But when we have other potential sources like the Tijuana River, that can actually confound these results. So what we did was we also sequenced the sewage coming from that plant, and we generated this fingerprint, which I talked about earlier. And then we looked for that fingerprint as we moved north away from the plant as well. So we kind of combined the two methods that we have to really get an idea of impact of this plume and how far reaching it is, and to try to separate the impact of this wastewater treatment plant from that Tijuana River source as well. So these results are ongoing, so I can't share any data with you yet today, but I can kind of give a shout out to the collaborative team that's made these efforts possible. The first being that we're working with a great nonprofit in Mexico, the Tijuana Waterkeeper, which is helping with our sampling efforts on the Mexico side of the border. We're also working with the Imperial Beach Lifeguards, and we're taking measurements both kind of where I am standing in this picture at ankle depth, and then we're taking measurements as well in the surf zone to see if exposures differ at those two locations. And then we're working with folks at the City of San Diego lab too. So this is big collaborative effort to kind of get these samples from this large stretch of coastline on the same day under these certain conditions to see what's going on with the sewage from that plant. So with that, I'll kind of talk about where we are going. So right now, all of the things that I talked about require that we actually take a water sample and then we go back to lab and process it, and then we have an answer and we can kind of report out and look at the data. Moving forward, the technology is actually getting there where we can kind of miniaturize and automate this workflow into something that looks like this, a briefcase, with the idea being that we could attach something like this to a buoy or something else and get these kinds of data directly to our cell phone. So NOAA, in collaboration with Monterey Bay Aquarium, is actually leading the way in developing these types of samplers. So soon, hopefully, we really can be like CSI and just kind of look on our phone and say, "Uh-oh, we've got a plume and it's hitting, beach X." And so with that, I'd just like to thank you all for tuning in, and I'd be happy to take any questions.
Thank you so much, Amy. This has been really interesting and exciting to hear more about your research, and just thank you so much for coming and joining us and sharing so much incredible information. We have some great questions. I'll just share with you. Jay brought up a couple great questions. Amy, in general, how do you determine where to take samples? It seems like it could vary a lot where you would take those samples. How do you figure that out?
Yeah, so that's a great question, Jay, and a lot of research goes into that. And it really depends kind of on the project. And so we do kind of a lot of investigative work to start with. We call it sanitary surveys. So we get like spatial information about potential sources, and maybe we look at monitoring data that the public health labs have and generate to look at historical trends and see if there are places where there are elevated levels, and then and if those elevated levels are occurring in particular seasons, and then we'll target our sampling kind of where we have an idea of that of where there are problems, if that makes sense. Yeah.
Great. That's helpful. I hope, Jay, I think we got a good answer there. Thank you so much. We also have another question from Jay. Are you noticing any trends that are affected by climate change or other factors and getting better or worse with that?
Yeah, so I didn't talk about it today, but that is another thing that's another kind of component of my research and something that I'm really interested in. And definitely we expect that climate change could kind of affect the frequency or the magnitude of storm events, which could definitely influence kind of runoff and a lot of pollution sources that we worry about that are associated with rain events. But we also have like emerging pathogens that can increase in concentration and kind of their geographic range because of climate change. So if you've heard about Vibrio in the news, that's something that I'm also working on. That's something we historically haven't worried too much about in California, but with climate change, we are seeing kind of that the geographic range is increasing, and that's a pathogen that's not from fecal sources. It's actually from the environment, but it can cause some nasty health impacts to humans.
Thank you so much. We also have a question from Lynn. Welcome, Lynn. She says, "Hi, Amy. On average, how many days a week do you spend out in the fields doing this great work?"
Yeah, not very many Yeah, not as much as I used to, I guess. Now I have lots of minions, but I am going sampling tomorrow because of all this rain that we had over the weekend. So let's see, maybe once a month, not as much as, not as much as I used to. So unfortunately, I guess that's how it goes. You know.
And then how, this one from Rick Lisan's hubby, how do you get the DNA from the bacteria? How do you actually extract that?
So we have all kinds of ways that we can do that. But what we do usually with like a water sample is that we'll filter it through a filter that has really tiny pores. So we can capture, bacteria that way on the filter. And then we kind of break the filter down. And then we use different chemicals to kind of break the bacteria apart and bind the DNA. We take advantage of specific chemical properties of the DNA to kind of bind it to a column and get rid of all of the other parts of the cell that we're not interested in and anything else that might be in that sample. Uh-oh, I might need
That's
water.
really helpful. Yeah, sure. Do you need to get water?
1 second, sorry.
No. And then please, everybody, keep you're bringing up so many great questions for Amy. We really appreciate it. Another great question comes from our board chair. And I know Christina, I think, is on as well, his daughter, who's really interested in this line of work and pursuing education in this. So hopefully we'll hear from her too, but welcome, excited you're here. What technical skills are most used in your research? Biology, data analytics? What are like the top maybe 3 to 5 things that you would say are the technical skills that are most used?
So, yeah, that's a great question. I think microbiology, for sure, you have to kind of understand who's there and what about the environment might affect who's there. But data analytics and data analysis skills are equally as important, if not more so, especially as we're kind of transitioning to like growing these things in lab, which is what we used to do. And now we're able to sequence things and we get a ton more data. And so it's really important to be able to kind of tease apart that data effectively. So I'd say the data analytics and data analysis skills are really important. But just because you have a ton of data, you need to be able to develop kind of clear hypotheses about who's there and why they might be there. Sorry, I don't know what's going on.
No, that's okay. It happens to the best of us, Sometimes we just get a little bit of a frog in our throat.
Yeah.
But we really appreciate it because there are lots of questions that have been coming in and very helpful information that you're sharing for us. So thank you.
One more thing. Oh, sorry. I was going to go back to the question.
Please.
Just to kind of continue on that path, I have to say everyone that I work with in this line, I don't think any of us have like a strict microbiology or molecular biology background. Most of us either came from like biological oceanography or environmental engineering fields that are pretty applied and allow for you to really kind of take a systems approach when you're looking at these types of problems. So.
No, that's really helpful. Maybe I can ask a little bit of an origin story. We're always very interested in origin stories at Discovery Cube because we really believe in a STEM identity and building the STEM identity for young people so they see themselves as scientists doing as scientists do. What was that moment for you when you said, I love science and I want to be, I want to go further with this? Like, what was it? What was your source of inspiration?
Sure. So, I've always loved the ocean and I have spent a ton of time in the ocean from swimming, as I mentioned, surfing, diving, kind of everything that I can do. But I actually went to college thinking that I would be a high school counselor and kind of was on that route. But I was really involved in surf and I started to collect water samples and I went to a big conference and I kind of got it in my head that, I could actually do this other thing that I thought was a hobby as a career. So I'd say kind of my involvement in Surfrider as a citizen scientist is kind of what originally drew me down this path. Yeah.
Well, that's really exciting. I know we were talking before about our Dive into Citizen Science program. So,
Yeah.
thank you for getting involved and helping us encourage others to do so. I'm going to go into a little more something specific, but a great question from Walkie Ray. How expensive, reliable, and quick are your DNA analyses? How do you do that in a fashion that gives you real data and is quick enough to make a difference in what we want to do?
So I kind of talked about two different ways that we use. One is we quantify specific DNA sequences that tell us about like particular sources. So that we can do fairly quickly, and SQUIRP has actually done in partnership with other local agencies, has done some pilot work to get those methods into practice for routine monitoring, because we can get an answer with those techniques the same day, which is a big improvement to what we do now to monitor for beach water quality, where we're getting an answer in 24 to 48 hours when we know that the water quality is a lot more variable on a much tighter time scale, shorter time scale, I should say. So those methods we can do fairly quickly. They are pretty expensive. I'd say, depending on like how many markers we want to look at. So if we are just curious about human, it's going to be cheaper than if we want to know, oh, is it human, is it gulls, is it cows, what have you. But it's around $100 a sample kind of per source type. Sequencing, the other thing I talked about, where we can kind of get a general idea of everybody that's there, and we can fingerprint just about anything that we want to fingerprint, like my example of how we're actually fingerprinting effluent from a particular wastewater treatment plant. Those methods are getting pretty reasonable in cost. And I would say per sample, it's maybe like $80, but those take a little bit longer. So you probably won't have a result. I'd say the fastest turnaround is probably about a week. And I can't see using those kinds of methods in routine monitoring right now. Because one thing that I didn't mention, kind of a disadvantage of those methods, is that we're not getting like quantitative information. It's all semi-quantitative. So it would be hard to use in like a monitoring capacity.
Just a little follow-up on that question. In something that you're doing, whether you're using sample or sequencing, how many tests do you have to run to get some valid data and data data points, data elements?
That's a great question. And anyone that's done any water sampling knows that water samples are highly variable, and we're just taking like a grab water sample usually, and that changes. If I took a water sample and then I immediately went back out and took another water sample at the same place, it would probably be wildly different. So to get a really good idea of kind of what your problem is and where it's coming from, like generally we say, that you have to sample over multiple seasons, and you want, I'd say 20 samples from a particular site, 20 to 30 over multiple seasons to start to get kind of a cursory understanding of what's going on.
Wow, that's great. And then I know we only have a couple more minutes, but I know before the session started today, I also asked you a question. It's a little kind of out, but I thought it might be a nice primer for our next speaker, which is going to be talking about COVID in kids, is the fact that they've been able to start to identify through, water pollutants and testing where COVID is, and been able to really use that. Just wanted to kind of get your insight in that and how that research is working and how it might be able to help us to deal with the crisis and pandemic we have now.
Sure. So sewage, for those of us that work in it, we think it's pretty great for this kind of thing, because it's almost like this awesome composite sample of a huge community. So if you imagine, say, if everyone in Orange County was to get a COVID test, or we were to require that, that would be a huge undertaking. But everybody is pooping, and that sewage is all going to the plant, and we have access to it. So from a scientist perspective, it's kind of this awesome composite sample where we can get an idea about an entire community without all the rigmarole of getting individual testing information and other things. But with that being said, there's a lot of promise in kind of using sewage and looking for COVID in sewage as a surveillance tool, but we're still very much in kind of the research stages. We're at the stage where we're trying to really, and my group is a part of this, trying to kind of standardize the methods between all of the folks that have jumped on board. So there's lots of academic research groups, there's lots of kind of governmental research groups, and then there's lots of wastewater treatment plants that are trying to all implement these methods to help us get a handle on kind of where COVID cases are increasing and where we need to kind of worry about those increases. So I'd say, first we want to standardize the methods. And then another thing we're trying to figure out is so that will give us a way to kind of compare, more easily between different places to kind of be able to actually transition just from generating data to using that data to generate real recommendations to the local health plans, if that makes sense.
Absolutely. That's
public health agencies.
That's super exciting too. Amy, I honestly think that I would have loved, and I hope that I get to talk to you a lot more, because I know even before we started, we kind of talked about diving into citizen science and some of the great ways we can continue to use your brilliant mind. So thank you so very much for joining us tonight and being just a total resource of the Scoop on Poop. That has been very, very interesting. And so just a big thanks to you for joining us tonight. And with that, I'm going to introduce our wonderful Janet Ray, who is the chair of our Innovators group. And she is going to have some closing remarks for us. Thanks again. And here's Janet. I think we can't hear you. There.
Sorry. Well, I'll start all over. Amy, I want to thank you for an excellent presentation. And I could hear my husband in the other room going, bravo, bravo, bravo. So I don't think any of us will ever forget Scoop on Poop. Thank you so much for great, a lot of information. And I hope that you can come back and to the Innovators again sometime. And I also want to thank everybody else for joining us this evening for the Cube Talks. This is our virtual science speaker series that we have every second Tuesday from 7 to 7:45, and we're just going to make that like you. Our hopes are that we're going to be able to meet in person this spring. But in the meantime, we have some fascinating topics and speakers scheduled for the next 6 months. And they are on the screen here. And the next Cube Talk is pertinent because in the pandemic, many of us are looking for information and research on how our children are faring in school. Dr. Dan Cooper, professor of pediatrics at UCI's Institute for Immunology and the founding director of the Institute for Clinical Translational Science and the program director for the UCI Clinical Research Center, consults regularly with the public health requirements of the state of California. Dr. Cooper is eager to share with you his latest findings. So please mark your calendar for Tuesday, December 8th at 7 o'clock. In this season of Thanksgiving, we want to share our gratitude to you for your involvement with the innovators and the support of supporting science and supporting our next generation. Thank you again, and good night. Have a great Thanksgiving. Yep. Yeah. You have to